Hi, well, welcome to another machine learning tutorial. In this video, I'm going to show you how to use a genetic algorithm for feature selection. So let's get started. Now, before I proceed, let me say that the notebook for this tutorial is available. So for those who want it, I will show you where to get this notebook, which has all the Python codes we are going to be using. All right, so let's quickly look at what a genetic algorithm is. And before I proceed, let me also say that for those who are beginners in machine learning and feature selection for that matter, I have some links in the description box that you can use to read more and get introduced to machine learning and feature selection for that matter. So let's get to look at what a genetic algorithm is. So I'll quickly shift to this slide for us to see. Now, with genetic algorithms, they are optimization algorithms that mimic the evolutionary process of living organisms to identify optimal feature subsets. So there are other algorithms that are also classified as evolutionary algorithms together with genetic algorithms. So these are things we'll talk about later on. Now, with genetic algorithm, it applies a series of evolutionary operators to search, and then the search is random based. It's a random search approach. So genetic algorithms can be parallelized as a way to improve them. And these are things we'll talk about later on. So this flowchart here lets us know how the genetic algorithm proceeds. So we input our data, initialize the population, compute fitness, then we go through this series of evolutionary operators, and then we do this until the stopping criteria is met. So it could be number of iterations or maybe a performance score or something, but usually we go through this several times, and then finally we get the best subsets at that time, and then we end it, and then we can use that feature subset for other things. That's what you need to do here. All right, so that's what we are going to do. So I will shift back to the genetic algorithm notebook here. So of course we need data, and so with data I have them here. We are using Wisconsin and Parkinson's. We will test all of them. So again, the links to these data sets are available. Just check the description box for them. So we are going to use them. The notebook is also available. So there's the notebook here. You can download and run it like I'm doing, or you can also run it directly from Google Colab. All right, so if you have Google Colab, you can just run. I have a tutorial that shows how to run Python notebooks directly in Google Colab. So just check the description box for that video as well. So that's what it is. Now back to genetic algorithm. I'll also quickly talk about data. So we have Parkinson's disease and then breast cancer. These are data sets we are going to use. And for the sake of beginners, I will say that the data sets here, you will need to process them to a format that you can feed to your Python codes. So I've already done that, and those are the ones that you can find here. But if you want the raw data as well as some additional information, check here and then you see them. Now with genetic algorithms, these are wrappers. So that means that we also need to have a machine learning algorithm that will evaluate the subsets that we select. So that's the genetic algorithm approach for us. So let's get started. Now we will start by running the code, some of the codes, by the way. So I also encourage you to get the notebooks and start. Then for things that are necessary later on, we will talk about them. So first of all, we have our genetic algorithm here. So we need to run it. These are Python code. This group of codes here import the necessary libraries for us. So I'll run, and then I'll go to this function. So I have a read file function that reads the input files. I'll talk a bit about them later and process them to a format that we can use. So I have my read files here. It needs train file, test file, validation file. So there's a function. I'll run all these functions. There's a function to generate or initialize the population. I'll also run it. I have a selection. So here I'm using tournament selection. I also have the crossover. I have mutation. I also have a function to evaluate the subsets of the chromosomes, the subset of features, or let's say chromosomes for that matter. And then the main algorithm that runs each of these functions and operators and give us our results or outputs. So I'll also run this. All right. Now I'll start here. So if I'm going to run the main algorithm, I need my train file, test file, validation file, and then some parameters which I need to set for the classical genetic algorithm. So The parameters we have: number of iterations, population size, crossover rate, mutation rate, right? So I need to also set them here. Okay, so for those who want a bit of explanation, I'll also leave some links that you can use to read more about the parameters in genetic algorithms. Now, these are ones that I've picked. You can also modify them to see if you get a better result from your own data. And of course, the data sets, because I'm reading them directly from GitHub, I need the link, and then I add some additional information to it, and then I'll run them. So basically, that's what we are going to do. Now, the data set, I'll say this. For example, in machine learning tasks, or let's say feature selection for that matter, this one data set. I think I'll open it for us to see. One of the aims of feature selection is to select a subset of the features, or let's say predictor variables, okay, a subset that gives a very good performance. So if you look at what we have here, there's a breast cancer data set where we have several measurements made for different patients, which are the horizontal. So each row represents a patient or individual or test subject. And then these are the measurements that were done for each of the patients. So you see that we have several measurements, or let's say variables. Or in machine learning, we also say features. So these are all features. And then we have the last class here, the last column here, which is class. So the class here represents, let's say, a label for each of the patients. Now for this data set, the metadata here indicates that the diagnosis section, which I've relabeled as class. So a patient or individual could be labeled as malignant or benign. All right. In machine learning, the computers may not understand these words, so we code them as values, as integers. And that is what I have done here. So 1 here represents malignant, 0 means benign. So you also have to know what numbers to use to do your coding. That's very, very important. All right, so that's the labels or class. All right, so that's fine. So the aim of this exercise here, which is feature selection, is to, instead of using all these features to build a model, we will now select a subset that will help us to build a good model. Now, usually with some of the data sets, especially those with lots of variables, some of these variables may not carry any information. Some of them are noise, some of them too are redundant. And so one of the aims of feature selection is to be able to select a relevant subset. Okay, and so that all this noise, all these redundant features, all these are eliminated, so that we have a smaller subset that can build a model. And when we do that, it also helps us to easily interpret and understand the models that we build. That's one of the aims of feature selection. So that's just by the way. So we are using genetic algorithms to select a subset of it that will give us a very good predictive performance. That's basically what we are doing in this exercise. So that's what we need to understand from this. So I'll get back to the codes and then I'll start. So I have defined here, I'm using the Wisconsin data, and then I need a train file, test file, validation file. It's also important to know that with the feature selection using genetic algorithms, we need a classifier because this is a wrapper approach. And so with wrappers, we will need a file for training and then a file for validation. So whenever we select a subset, we evaluate with a validation file. And then when we are done and we have our final subset, we test with an independent data set, which is now the test data, which is the test file here, this one here. So that is why I have 3 files here. Some techniques will also merge these two, train file, validation file, and then use what you call the k-fold cross validation. All right, so that's something you also have to take note of here. So that's what we have here. So I have my data, and I have my train file, test file, validation file, and I also have my parameters, which we need to set. Okay, so n iteration, so that means that how many rounds of this evolutionary process should be done. Here I set it to 10. Population size, I will explain later on, but we need to initialize with some data. And how many of such data should we initialize? I set it to 20. You can also set to 10, and that's also fine. It depends on you. All right. We also have mutation rate, which is this one, and crossover rates. Again, there are some links I will leave for you to read more about these things. You need to understand these things also before you can run and appreciate these codes. So that's what we are going to be doing here. All right. So now let us run the algorithm, the genetic algorithm. So I'll run it and then we see what we have. So here I set it to 10 iterations. Right. So it will run for 10 times. Now when it's running, at each iteration, the best result will also be displayed here for you to see. And then we do it and then we observe. All right, so I'm going to run for 10. So again, we have your selection here. When we run the algorithm, we start once we generate the population, which is here, like in the flow chart, we perform a selection. So selection allows you to select a subset that we now use for the reproduction. So here the reproduction is where we apply the evolutionary operators, i.e. crossover and then mutation. And then once we have them, the offsprings we have replace the old generation, we evaluate them and we get the best organism. That becomes what? The best in the particular iteration. And then we go through a series of evolutionary steps again. We do this for several number of times, and once we are done, the overall best chromosome, we now get it here. Now with the initialization, which I'm going to talk about, which is here, we need to represent the features as what? Chromosomes. I will shift back to this one here. I think it will be here. Let me just shift here. So we represent everything as this particular form here. So these are the populations. We initialize with this. There's a representation. So here it indicates that there are 5 features in the data, and then out of these 5 features, 3 have been selected. There's a binary bit. So 1 indicates that the feature has been selected. 0 means it has not been selected. So this is how we represent it, and then we run. All right. So once we do that, then we proceed. So I also quickly talk about the crossover, which is this one here. With crossover, we are going to exchange genetic material, okay, between 2 individuals, which I call parent 1, parent 2. So this is parent 1, this is the genetic material. This is parent 2, genetic material. So when we exchange, usually we have to set a particular point. So that position is where the crossover will take place. So here, that's where this red line is. So this point here indicates that this is the point. So that means genes on the left and then genes on the right, there's going to be an exchange. The same with this one here. So here, what we see that in parent 1, okay, we see the first offspring maintaining the genes for parent 1 and then taking the second half, that is this one, the genes in the second half of parent 2. When you go to offspring 2, it maintains the first half of parent 2 and then takes the second half of parent 1, which is this one here. So that's why we have 2 offsprings here. That's for the crossover. And then for mutation, we are basically flipping the bits. So if it's 1, it becomes 0. If it's 0, it becomes 1. So here, for example, this is an individual. When it undergoes mutation in this particular position, that means it's flipped to 0. That means the gene has not been selected. Individual here is 0, it's flipped to 1. That's also a mutation. Now, this position here, the classical genetic algorithm is random. All right, it's a random value that is generated, and if it meets a certain condition, then that position will be mutated. There are approaches to make it a guided approach, but these are things we'll talk about in another tutorial. So that is just an overview. So let's run the codes and we can talk more. So I'll get back to the Python codes and I'll run the algorithm. So let's run this. Again, down the notebook, the link is in the description box. All right, so take note of that. Okay, so let's run it and then let's see what we get. And also take note that depending on the data size and the information available, it will take some time. But with this data set I'm using, because I don't have much information, it will not take much time. It will just be running on still a moderate laptop or computer, so no need to worry. Okay, so you can see it's running. We have generation 0, 1, it's ongoing. So here we see generation 0, which is the first iteration. The best feature subset had a score of 0.9037 and it's 8 features. Now when we go to generation 2, it's the same score, 9. When we go to generation 3, the same score, 15. Okay, it appears as if it's increasing and then it decreases again. So this will go on for several times until we get the optimal subset, or until the termination criteria is met. So in this case, the termination criteria is the number of iterations. So once the number of iterations gets to the limit that we set, which is 10, it will stop. And then it will evaluate the subset on the test data set for us to see the performance of the selected subsets. So that is what we are waiting for here. So this 10 that we see here, you can also set it and then see. Now here you will see that in the training phase, when we were doing the feature selection, the score was 0.90, and now when we got here on the test data set, it's 0.88. Ideally, it should be similar, but then with this process, which is random search, sometimes some information, some algorithms, or let's say with a random search, what happens is that there's the possibility that the search will proceed in a region which will give a suboptimal result. It doesn't take into consideration your past discoveries. All right, so this can be a drawback of the genetic algorithm. But of course, when you run it for several iterations, studies have shown that you get a good result. So this is just a demonstration. So you can increase it to, let's say, 100 or 20, and then just test them. All right, and then see what you get finally. That's what you should also take into. So once you get this, it's important that you now extract that feature set, and then you can use that to build your model, or use that to complete your next plan. All right, so sometimes once you identify the subset, you want to do further tests, look at relationships, interpret it. These are things you can also do before you actually take this subset into production, or to put it in a production model. These are things you have to look at here. All right, so if it's disease prediction, like in our case, then the subset we have, okay, that subset that we have, which is 14, that means that you can extract that information from here. Let me show you. You can extract that information from here, and that means that subsequently, whenever you want to make your predictions, instead of spending money and time to take all these measurements, all these variables, you just focus on just the 14, because the 14 that we have here are giving you good results. Okay, so these are things you can also think about when it comes to genetic algorithms. All right, now with genetic algorithms, that's the classical genetic algorithms. They have some drawbacks, so I thought I'll also talk about that briefly. So for the drawbacks, one of the drawbacks is that it has high computational cost. Because it's a random search, you need several iterations to be able to get a good result. And also you need to tune the parameters. It can be hard and demanding. And then research has also shown that the classical genetic algorithms lack diversity. So it's not able to do a proper exploratory work. And also it's accessible to local automatic trap, and there's also the issue of premature convergence. So these are things you need to always consider. Nevertheless, there are techniques that can be used to improve genetic algorithms. And so when we talk about the techniques, there are those that use para-search, and there are also those that tune the parameters. You can also use immigration techniques, and there are also those that use adaptive techniques on mutation, crossover, immigration, or maybe use a set of probabilities instead of purely random, just to improve the search. So these are things we can look at. So for those who want to also implement some of these techniques, you can also contact me. I have some codes actually that implement some of these to improve the genetic algorithm. So that is all for genetic algorithms. And again, for those who want the notebook, just visit my GitHub repos. I'll put all the links in the description so you can get your notebooks and run them yourself. All right, so that's all for this tutorial, and I'll see you in the next session.